cloud. Okay. Okay. So in this chapter, okay, as I told you that this is a pure, molecular biology chapter. Okay, not in biochemistry. We try to know everything. We should know the molecular basis of everything, right? So in this chapter, the focus is basically on the structure of the nucleic acids. Okay. So because nucleic acid is also one of the biomolecules, the way we discussed about protein is a biomolecule in our body, lipids are the biomolecules in our body, carbohydrates are the biomolecules in our body. Similarly, nucleic acids are also considered as the biomolecules, right? Because our DNA and RNA, right? It's made up of the nucleic acid. It's made up of nucleotides, right? So it's considered one of the biomolecules in our body. Okay. So let's start. Okay. So in this chapter, as I said that this chapter is totally based upon the structure of the DNA, RNAs, about the different types of the RNAs. Okay. We'll be also talking about what are genes. Okay. And then in this chapter, we'll be also talking about that in Wednesday's class, we might talk about the DNA replication. Okay. Then transcription and translation is reserved for the next chapter, for the next week's chapter. Okay. So in this chapter, the focus is mostly on structure of DNA, RNA. Okay. Different types of the RNA. Okay. And then the transcription and and then the replication of the DNA, how the DNA replicates itself, and what are genes. Okay. And how things are repaired in our body. Okay. If something goes wrong in the body. Okay. And how we can amplify the DNA. We'll talk about the PCR also in this chapter. Okay. This is an interesting chapter. Okay. But we are not going very deep in this chapter, because if you talk about replication itself is like, 3-4 hours lecture. Okay. So we kind of, this is like a tidbit of everything. Okay. So we're not going very deep into everything. Okay. Because as I told you that if you talk about replication, okay, replication itself is a very long topic, which you can teach for hours together. Okay. But here we have kind of, not the overview, we're going a little deeper, but not very deep at the same time. Okay. So that's what you can expect in this chapter. Okay. So let's start the chapter. Before we start, I want all of you to understand that, because what we are going to talk about, it's a part of a genetics, okay? Human genetics, for example, right? So can anyone explain me what is genetics? Genetics is itself a topic. It's the way you have your anatomy, you have your physiology, you have your biochemistry, right? Genetics is also one branch of the science. How will you define genetics? Nobody? You can switch on your mics if you want to, if you're comfortable enough.
Something that's inherited from your parents.
Very good. Absolutely. Thank you. Anybody else? How will you define genetics? The study of the gene patterns, sequence of nucleotides, study of the genes. Very good. Absolutely. Yes. Anybody else? Okay. So genetics is basically, as you rightly said, that the study of the genes, the study of the heredity. Absolutely, Dana. Absolutely. Right. The study of the heredity, the study of the variation. You might have seen that, you look similar to your mother or father, right? But you're not exactly same as your mother or father, right? There's a little kind of variation in an organism. Okay. So that is your genetics, right? When we talk about how traits, what are traits? You know what are traits? Yes. Can you give me any example of a trait in your body? Eye color, absolutely. Blonde hair, curly hair, right? Your height, blood type, A, AB, O, skin color, dark tone, light tone, right? Or resistance to any diseases. Okay. So all these are considered as, you have a long nose, long face, short face, right? So all these things are considered as the traits. So in genetics, we talk about usually how these traits are transmitted from father to the offspring, from parents to their children, okay, or from grandparents to their grandchildren. Because sometimes you might see that, oh, baby's having green eyes, okay, exactly like his father. That means traits have passed from father to the baby. Oh, exactly. His nose looks exactly like his grandfather. Okay. Or his hair is very curly like his grandfather. So traits were passed from grandfather to his son and from his son to the next offspring. So basically, genetics also talks about the inheritance of the characters from one generation to the other generation. And in our world, millions of species, and whether they're animals or human beings, right, they have millions of traits, right, and these traits are transmitted from one generation to the other generation. Okay, so genetics as a branch of the science, it tries to explain how these traits are transmitted. Okay, it also tries to explain how exactly, what is the composition of these traits. Okay, and it also talks about the genes. Okay, in this chapter we'll define a gene. What is a gene? Okay, does anyone know what is a gene? What is the difference between DNA and a gene? The question says that what is the difference between a DNA and a gene? How will you define a gene? Strand of DNA, okay, small part of the DNA, but what is happening with that small part of DNA? Mm-hmm. Gene is a part of DNA. Absolutely, that is what I was looking for, right? So if I say that this is my DNA, if I say that, sorry, if I say that this is my DNA with the, I'm sorry, this chapter needs immense writing, but the problem is, I'm not able to write this pen. Okay, adenine, thymine, guanines, for example, cytosine. Okay, cytosine, adenine, guanine, right? If this is my entire DNA, which is very, very long in my body, but gene is only that part of the DNA which has some sense. Because, like, most of your DNA is just the repetitive sequences. We call them palindromic sequences, like adenine, adenine, adenine. They're just repetitive sequences, like, vague DNA, which has no nonsense DNA, which has no role in our body, right? We call them palindromic sequences. But only certain parts of our DNA which codes for a particular mRNA, okay, and that mRNA codes for a particular protein, that part of the DNA will be called as a gene. So your entire DNA cannot be gene, because I told you, like, most of your DNA, 99% of your DNA is a junk DNA, right? Just the repetitive sequences, adenine, 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 thymine, 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 guanine, 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 cytosine, 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 which has no role to play in terms of the protein formation, right? So very small amount of DNA codes for a particular mRNA called as transcription, which that mRNA codes for a particular protein, which is expressed in our body and shows a different kind of, action in the body. Are we okay? Right? And the traits, for example, you might have studied in your high school, okay? If you remember, I hope you know the name of Mendel, right? Mendel is called as the father of genetics. Okay? The father of classical genetics is the Mendel. You might have studied all that in your high school, right? It was Mendel who, for the first time, basically said that who identified these traits and said that there are factors in our body which control these traits, right? Eye color is a trait. It's back there in your DNA that you're going to have the brown eye color. Your hair is curly. It's right there in your DNA, right? So that you can produce more cysteine, a protein, and you can have more curly DNA, curly hair, right? So everything is written in your DNA, everything is there in the DNA, right? And based on that is, it's expressed in your body, right? And that's how the traits are controlled. So Mendel at that time just said that there are factors, okay, which control these traits like height, eye color, blood type. But later on, we gave that, the name factor was replaced by the genes. Now we say that genes control the traits in our body, okay? And these traits are basically present in the genetic information, which is carried by a special molecule called as the DNA. So having this long discussion was basically so that I can introduce the term called as a DNA to you, because the whole chapter is based on the structure of the DNA and the replication of the DNA. Right? So every living creature on this earth, except few viruses, they have the DNA back in their, either in the nucleus, if it's a eukaryote, if it's a prokaryote, it's scattered in the cytoplasm. Okay. Now, if I ask you, where is DNA located in our body or in our cell, to be more precise enough? Where can we find DNA in the cell? In the nucleus. Do we have DNA in the cytoplasm as well? Do we have DNA in the cytoplasm as well? In the form of, very good, in the form of? No, mRNA is not the DNA. mRNA is mRNA. It's the RNA. It's different from the DNA. Right? Mitochondrial DNA, absolutely. So you have to remember that DNA is basically confined within the nucleus, right? DNA is usually present within the nucleus in the chromosomes, right? But a very, very small amount of DNA is also present in the mitochondria in the form of mitochondrial DNA, which we learned in chapter number 3 last semester. Mitochondria has its own DNA called as a mitochondrial DNA. Okay. Now, when we talk about RNA, okay, where is RNA located? We'll not discuss about different forms of the RNA. That is a different topic. We'll discuss about that. But let's try to find out, the basic things. Where is RNA present in the body? Whether it's
outside of the nucleus.
outside the nucleus. What about within the nucleus? Do we have any RNA within the nucleus? Anybody?
Yes.
Can you name the RNA which is in the nucleus? So basically, you have to say that RNA is the one which is present not only in the nucleus, RNA is present in the nucleus as well as it is present in the cytoplasm. Okay, it's present in nucleus as well as in the cytoplasm. Because if you see that once we discuss the DNA, because DNA forms the mRNA. What is mRNA? mRNA is an RNA, right? So we find that mRNA in the nucleus first, then it travels from the nuclear pores into the cytoplasm for the protein synthesis. But initially it's formed in the nucleus, right? Ribosomal RNA, we spoke about that in chapter number 3. Nucleolus, right? The RNA assembly, right, takes place in the nucleolus. So that means it is there in the nucleus as well as it is there in the cytoplasm. So ribosomal RNA, it moves from the nucleolus to the nucleus and then into the cytoplasm, right? Then it forms with the proteins, it gets joined and forms the ribosomes, the ribosomal RNA, right? The rRNA. We'll talk about all these in detail. This is just an introduction. Okay. So that is found in the cytoplasm as well. Clear? And the structure of the DNA. I will not go into the DNA history, the DNA, how DNA was discovered. It has a beautiful history, but as the chapter is already very long, I cannot go into it, right? So DNA is basically, it was discovered by Watson and Crick. The structure of DNA was given, in 1940s, okay, by Watson and Crick. So that solved all our problem, that's how we came to know that the DNA is double-stranded, contains nucleotides, it has 5' 3' and all those things, Okay, so that was just the introduction of the chapter, and we move on now to the nucleic acids. Okay, so when we talk about the nucleic acids, as I told you that nucleic acid is a biomolecule. The way we have your proteins, lipids, carbohydrates, nucleic acid is also one of the biomolecules. And we have two types of nucleic acids in the body. We not only have DNA, we also have the RNA. Okay. And what is the full form of DNA and the RNA? Can anyone tell me what is the difference between, let me see how many of you know the difference between DNA and RNA? What is the full form of DNA? And you can even switch on your mics if you want to. And what is the
DNA
basic
is
difference between? Mhm. Please go
deoxyribose
ahead,
and
dear.
RNA is ribose.
Deoxyribose. No, it's
Sugar.
the deoxyribonucleic acid. This is the full form. There's a word N as well. Nucleic acid shows us that DNA is totally confined to the nucleus. Okay, except the mitochondrial DNA, which is present in the cytoplasm. So don't forget the word nucleic acid as well. The nucleic tells us that it's present in the DNA mostly, in the nucleus mostly. Okay, so very good. So missing O in the DNA, deoxy, okay, nitrogen bases A and U. Okay, very good. Right? So you can say that DNA and RNA are both nucleic acids, right? But the basic structure is similar, but yes, there are a lot of exceptions between DNA and the RNA. Okay. So before we begin, you should know the basic difference between DNA and the RNA. So if you say DNA and the RNA, we know that if I put a line over here, okay, so if we say this is DNA and this is RNA, the first difference between the two is DNA is double-stranded, RNA is single-stranded, right? And then what is present in the chromosomes? Do we have RNA in the chromosomes? Along with the histone proteins, we spoke about chromatin in PHSF last semester. Do we have RNA in chromatin? No, of course not. Chromatin has only histone proteins and the DNA. So DNA is present in the chromosomes, RNA is not present in the chromosomes. Right? In DNA, we have bases like your adenine, we have thymine, we have guanine, we have cytosine, right? But in RNA, we have adenine, we have no thymine in RNA, we have uracil, we have guanine, we have cytosine, right? Clear? Yes. Okay. And DNA is responsible for storing the information, right? Storing the genetic information, isn't it? How your hair is going to be, it's going to be straight or it's going to be curly. Okay. Are you going to be dark or you're going to be light colored, your skin tone, right? So that information is stored in the DNA, right? But RNA does not store the information. Rather, RNA codes for the information, isn't it? It codes which amino acids are going to be there in the protein, right? Rather than, yeah, it synthesizes the protein. Absolutely, Mohammed, right? It forms the protein, synthesizes the proteins, right? It acts as a messenger between, for example, mRNA is a messenger between DNA and the ribosomes, right? And then one more difference is DNA is self-replicating. That is such an important point which you should not forget. You should know all these differences, right? Self-replicating. We'll discuss at the end of this chapter how DNA is going to replicate, right? But there's no question about the replication in the RNA. RNA is totally dependent on DNA, right? RNA is formed from it's the DNA which forms the RNA based on what is the need in the cell, right? DNA is formed, RNA is formed from the DNA. Okay. And last but not the least, the DNA has the base sequences. For example, guanine pairs with the cytosine in the DNA. Adenine pairs with the thymine. Okay, but while as the base pairing in the RNA is the adenine base pairs with, all these things are important in terms of the questions. This is all these things we're going to discuss again and again, as I will progress in this chapter. Okay, so this is just an introduction, and one by one I'll be discussing all these things once again. Yeah, the ribose and the deoxyribose, absolutely, yes, yes. So adenine base pairs with the uracil, isn't it? And guanine base pairs with the cytosine here, right? And of course, we forgot about the sugar part, right? Thanks for reminding. So sugar is basically in the DNA, in the RNA, both of them have the pentose sugars, right? But in the DNA, we have the deoxyribose sugar, while as in the RNA, we have the just ribose sugar, okay, which we'll be discussing in a while. We're at position number 2, right? We have OH, but in DNA, we don't have oxygen. We just have the hydrogen at position number 2. We're going to discuss all that now in detail now. Is it clear? This was like, the basic things which you're expected to know, because, you have studied all this in your high school as well. Okay. So now whether we talk about RNA or DNA, okay, we say that they are polymers, okay, which are made from the nucleotides. What does that mean? That means if we talk about proteins, the way you discussed in proteins are made up of amino acids, right? Lipids are made up of fatty acids, right? Similarly, your RNA and DNA are made up of the monomers called as the nucleotides. Okay, are we okay till now? And these repetitive, so that means any DNA or the RNA is a long chain of the nucleotides. We also call them as a polynucleotides. Okay. And now what is this nucleotide made up of further? Nucleotide is further made up of a base, a nitrogenous base, which can be any adenine or thymine or guanine or cytosine or uracil, right? And then we have a sugar, which is always a monosaccharide, which is a pentose sugar, okay, which is, as we discussed, it's a deoxyribose in DNA and ribose in RNA, okay? And then we, of course, have the phosphate. So these three things are present in a nucleotide. Are we okay till now? Any questions till now? Yes, ladies and boys. Okay. Alhamdulillah. Great. If you don't understand in between, please intervene and stop me. Okay. So that I can re-explain it to you. Okay. Don't worry about that. So what are the two important functions carried by the DNA? We already spoke that DNA carries the genetic information. It stores the genetic information. Okay. It's called as a blueprint of your life, right? But then what is the function of the DNA? Other than storing the information, DNA has a very important function called as the DNA replication, right? So if the DNA will not replicate, for example, from one DNA, it becomes what? It replicates and forms. We discussed the semi-conservative model of DNA, right? In PHSF last semester, right? From 1 it becomes 2, from 2 it becomes 4, from 4 it becomes 8, right? It has a capability of forming the exact complementary replica copy of itself, right? And that new DNA is further packed in the cell, right, during the cell division. So there will be no cell division. Our cells will not divide if the DNA will not replicate. Can you imagine? So we will not be able to grow if our DNA will not replicate. Don't you think it's an important function for the DNA? Right? making its own copies, right, is a very, very important property of the DNA. It's one of the most important property of the DNA, right? So guys, whatever I'm telling you, each and every point, just pay attention to it, because the questions will be based on that. Okay, so don't miss a single point, whatever I'm saying. Okay. And the second most important function of the DNA is, rather the third most, first was, storing the information, right? The third most important function of the DNA is the protein synthesis, right? Expressing a particular mRNA. Okay, so if there's no DNA, if the DNA will not form mRNA, right, by the process called the transcription, then no protein will be formed, right? So DNA has the information on it. It has basically a recipe on how to make each and every protein in our body, right? So that is why it is very, very important in terms of protein synthesis. Shall I move to the next slide? So who will answer this question? Is RNA present in the chromosomes? Do we have RNA in the chromosomes? No, right? We already discussed that. RNA is not present in the, it's only DNA and the histone proteins, right? Which is present in the chromata. Very good. Okay. Moving on further, let's discuss about the sugars now, okay? Because we discussed that in a nucleotide, okay, we have a sugar, we have a base, and we have a phosphate, right? So let's discuss the sugar, okay? Sugar, as I already introduced the concept to you. I already told you that the sugar present in the DNA or the RNA is a pentose sugar. Okay, so is it, can anyone tell me what kind of ring is this? I think we discussed that in carbohydrates. It's a 5-membered ring, I know that, but what is this 5-membered ring called?
Furanose. Furanose.
Absolutely, mashallah, very good. It's a furanose ring, right? So the sugar present in the DNA is in the furanose form. Very good, absolutely. If you see, there's an oxygen over here other than carbon atoms, okay? And then if I ask you another question, this question was, most of the people made this question wrong, which was about the anomeric carbons. I will discuss that in Wednesday's class. What kind of sugar is present in the DNA? Is it deoxy? Is it D or the L form? For example, is it D ribose or is it L ribose? Or is it deoxy D ribose or deoxy L ribose? D. Yes. If you remember, I told you that we do not have L forms of sugars in our body. The naturally occurring sugar in our body is only the D form, right? So there's no L form of the sugar. So the question says that the sugar present in our DNA, what is it other than knowing it's a pentose sugar, it's a furanose form? You have to know that it is it's only the D form of a sugar. So it can be a D ribose or it can be a deoxy D ribose. Okay. Now the difference between the two is, as you can see here, this is the sugar present in the DNA and this is a sugar present in the RNA. Okay. If we start naming them, from here, okay, we'll see that at position number 2 in the RNA, we have OH. Okay. And if this structure is given to you, you should be able to identify which sugar is present in the RNA and which one is present in the DNA. And there's no oxygen at position number 2 in the DNA, right? That is why the word deoxy means, that means no oxygen is in the DNA. Okay. Now that was about the sugar. Okay. Moving on further, what are the bases present in DNA? As I already explained you, we have adenine, thymine, guanine, cytosine, right? These are the bases present in the DNA. But if we need to categorize them, we say that the nitrogenous bases are either pyrimidines, okay, or they are the purines. Okay. All you need to do, you don't have to remember the structures of the pyrimidines and purines. I know that this question will come up. No need to memorize the structure of the pyrimidine and the purine. Okay, but you should be able, as I said, that you should be able to identify. Okay, for example, if I give you a structure like this, okay, you should be able to know that this is a pyrimidine, which is very easy to remember. That if you see here, the single ring compounds are your pyrimidines, right? As you can see here, the double ring compounds are your purines. So you should know which bases are purines and which bases are pyrimidines. Adenine and guanine is a purine, okay, while as cytosine, thymine and uracil are all pyrimidines. You should be knowing all this, okay. And if you compare the cytosine and the uracil, cytosine and thymine and the uracil, this and this. Can anyone tell me what is the difference between thymine and uracil? But just looking at the structure. CH3, yes, Nora, absolutely, absolutely. At the position number 5, okay, in the thymine, okay, we have, so you should be knowing this information. At position number 5 in the thymine, we have a methyl group. However, there's no methyl group at the position number 5 in the uracil. Rest the structure is similar. Okay. Okay, this is the same thing what I explained it to you, which one is a purine, which one is a pyrimidine. So you should be knowing which one is a purine, which one is pyrimidine. If you're asked if it's what is uracil, is it a purine or it's a pyrimidine, right? And if you're given a, for example, if you're given a structure like this, like a double ring, you should be able to identify is it a purine or it's a pyrimidine. The only thing to remember here is purines are, we have double ring compounds as purines, okay? While as the pyrimidines, we have a single ring. That's the best way to remember them. Okay. That was about the bases. Okay. Now, the bases are attracted. Now, there's a complementary base pairing in the DNA. Okay. That means guanine will always base pair with the cytosine. And for example, adenine will always base pair with the thiamine, right? There's a complementary base pairing here. And between the guanine and the cytosine, there are always 3 bonds present. Remember all these informations. And between the adenine and the thiamine, okay, how many bonds? Does anyone know? Did you study all this before? Okay, great. Okay. So they are the hydrogen bonds. Okay, in chemistry, they are the hydrogen bonds are the weaker bonds. Okay, so so they're the weaker bonds. But since there are many hydrogen bonds present in the DNA, that stabilizes the structure of the 2 strands of DNA. But don't worry, I'll come back to this once again as I'll show you the entire structure of a DNA strand. So don't worry about that. Okay, this is just, the introduction. We're going to repeat the information once again as we discuss the entire structure of the DNA. Okay. Now, what is a nucleoside? Again, a very important thing. You should be knowing the difference between a nucleoside and a nucleotide. Does anyone know the difference between the two? Nucleoside is, and what is nucleotide? No? Okay, don't worry. Okay, it's okay. I thought you would have studied this in high school. That's okay. I will explain, okay? So now pay attention. Nucleoside, when we say the word side, look at the word, or look at the spelling over here. When you see only the side, okay, nucleoside only contains your base and your sugar, okay? So when we have base, okay, a nitrogenous base plus a pentose sugar, okay, these two are called as a nucleoside. Nucleoside. So this is very important. Please pay attention. And then when we say nucleotide, okay, your DNA and the RNA are made up of many nucleotides, not nucleosides, right? Nucleotide. So in the nucleotide, we simply need to add a phosphate to these two. So we have a base in a nucleotide. We have a sugar. So that means you have a nucleoside first. And simply you add a phosphate to it. It becomes a nucleotide. Okay. Not always mono, they can have 2 or 3 phosphates as well, but nucleotides usually have only one phosphate. Okay, the nucleotides present in the DNA. We'll talk about that in a while. Okay, so what is a nucleoside? Remember, simple, base plus sugar is a nucleoside. Okay, if you look at the structure over here, this is the structure of the pentose sugar. Okay, this is the pentose sugar over here. Okay, and if you see here, now I want all of you to pay attention. You remember what is an anomeric carbon, right? This is an anomeric carbon for the pentose sugar, right? This one, this carbon, okay? And remember this point is very important for all of you. Please pay attention. Now, we said that in nucleoside, you have sugar and base, that is it, right? So this is my sugar and this is my base, right? So sugar and base is, you have to remember that base is always connected to the anomeric carbon. That means carbon number 1, okay, through a bond called as a beta N glycosidic bond. This is a glycosidic bond, okay? So remember that the base is always attached to the pentose sugar at the anomeric carbon, which is the carbon number 1, okay? And remember, for example, what is this bond called? It's called as a beta N glycosidic bond, okay? If you see here, what is it? Is it a purine or is it a pyrimidine? Just don't tell me what kind of base it is. Just tell me, is it a pyrimidine or a purine? The structure which is right over here, this one, a single ring
Pyrimidine.
structure. It's a pyrimidine, right? So remember, I'm going to repeat it once again. So if you have a base in the form of a pyrimidine, okay, that means if it's cytosine, thiamine, or uracil, right, they are connected, okay, at the position number 1 of that base. Okay, so N1 of pyrimidine is connected to the anomeric carbon of the sugar. Okay, but just in case, if it's not a pyrimidine, if it's a purine over here, okay, if it's a purine over here, that means if it's an adenine or a guanine, so it is not the nitrogen 1 at that time, it will be the nitrogen 9, N9. This is the only thing which you need to remember here. I repeat, nucleoside is a sugar plus base. Both of them are connected by a bond. Remember the bond, right? The bond between sugar and the base is the beta and glycosidic bond. That is it. But then I told you that if the base is here, pyrimidine, that means if I have cytosine, thiamine, or uracil over here, then it is the N1 of my pyrimidine. But if it's a double ring, okay, it's a double ring, right? I showed you the purines, right? I showed you the purines over here, right? So if it is a purine, then this nitrogen will be attached to the sugar. This is the only thing which you need to remember and pay attention to this. Forget about structure, remember the names. Okay? Are we okay about it? Do you want me to repeat or it's okay for everyone? So N9 of purine, okay, N9 of purine is connected to the anomeric carbon of the sugar, but N1 of pyrimidine is connected to the anomeric carbon of my sugar. Any questions? I'm going to erase all this so that you can have a clear slide and clean slide. No questions? Okay, you remembered it, right? Okay, very good. Now, the second thing which I wanted to, now again, I want you to pay attention. Yes, Hussein, I will repeat once again, don't worry. I will sum it up, okay? Just pay attention what I'm trying to say now. Okay, I said that N1 of pyrimidine is connected to the anomeric carbon of the sugar, right? C1 of the sugar. But if it's not a pyrimidine, if it's a purine, then here you will have N9. That is all you need to remember. Okay, the difference between the two. And then I told you that nucleoside is a sugar plus base. The second important thing which I want you to remember in this slide is that, do you see here 1 dash, 2 dash, you know what is a dash, right? Like this, the prime number dash, right? We call them, yeah. Now, this is a very important thing to remember here, okay? Now, whenever you will see the sugars are numbered, whenever we talk about nucleotide or a nucleoside, okay, whenever these sugars are numbered, you will see that they're numbered as 1 prime, like 1 with a dash. So the moment you see 1 with the dash or 2 with the dash, immediately it should come in your mind that, oh, this is a numbering for the sugar, right? But when I number, I'm going to erase this. But when I number my bases, for example, I don't use any dash here. I simply use 1, for example, then this will be 2, this will be 3, this will be 4, this will be 5, this will be 6. So I don't use any dash when I number my bases. The reason for that is to avoid any confusion between numbering of the bases and the numbering of the sugars. Okay, we use the prime number dash only for the sugars whenever we are naming the sugars. Is it clear? Everybody understood? So if I say, for example, let's say I have phosphate attached here. Let's say my phosphate is attached at position number 5 here, right? Phosphate is attached to position number 5, for example. How would I say that? I will not say that it is the phosphate is attached to position number 5 of the sugar. I will simply say that phosphate is attached to the 5 dash carbon of the sugar, right? So the moment you see 5 dash, you should immediately remember that it is about the sugar, not about the base. Clear? Should I move to the next one? Okay, very good. Then nucleotide. I explained you here, nucleotide is a sugar plus phosphate and the base, right? It's simply when you add a phosphate, when phosphate is esterified to the sugar, we simply say that it is a nucleotide. So nucleotide has a sugar, which is this one, okay? Nucleotide has a base, which is this one. As you can see over here, is it a purine or a pyrimidine? Double ring base. It is a purine, right? Absolutely. And nucleotides also have the phosphates attached to it. As you can see here, this is one molecule of phosphate. Okay. This is another molecule of phosphate, and then 3 molecules of phosphates are attached to it. But that's not the always case. Okay. So in the nucleotide, you can have 1 phosphate. In the nucleotides, you can have 2 phosphate. In the nucleotide, you can have 3 phosphate. But remember, the nucleotides which are present in our DNA, they have only how many phosphates? 1 phosphate. So the nucleotides are the building blocks of DNA and RNA, and these nucleotides have a base, They have a pentose sugar and they just have one phosphate. Okay, I will show you this is a nucleotide. If you see here, this is a nucleotide. Look at this. It has a base, it has a ribose sugar, and it has one phosphate. So this becomes my nucleotide present in the DNA or the RNA. Are we okay about it? Did you all understand? What is the difference between general? So generally we say nucleotides have sugar, phosphate, and base. But when we talk about the nucleotides present in the DNA or the RNA, they just have the monophosphates. So that's why we call them sometimes we call them nucleoside monophosphates. Yes, Noora, I'm coming to that point. We'll go step by step, one by one. I'm coming to that point. Okay. And remember that this phosphate, which you see over here, this phosphate can be attached to the sugar either at the 3' position of the sugar, or it can be attached also at the 5' position of the sugar. It can be both the things. Remember this point as well. Okay, so you can say that if you see the sugar is the phosphate is attached to the pentose sugar, and it can be attached at the 3' position, and it can be attached at the 5' position. If you see here in this one, it is attached at the 5' position. Look at this, right? See, let me get a red pen, red color. See here, it's attached at the 5' position, right? But if you see here in this nucleotide, it is attached at the, let me erase this, it is attached at the 3' position. So this is also the point to remember, that sugars can be attached to, the phosphates can be attached to they can be esterified to the sugars either at the 3' position or the 5' position. Okay, so if you see here, this is so deoxythymidine 3' monophosphate. Okay, for example, the question says that what does this 3' refer here? You will say that 3' means that the phosphate mono, that's one phosphate, is attached to the sugar at the 3' position. The moment you see this small comma over here, we call it a prime over here, right? Prime number over here. Okay, so this is deoxythymidine. So when you say the word deoxy, that means you know that this is it doesn't have the oxygen at the position number 2. Monophosphate, that means it has only one phosphate attached to it. Okay, now somebody asked me a question. I think, yeah, Noora asked me a question. What about when we have 3 phosphates? Yes, we have nucleotides with the 3 phosphates. If you see here, this is a nucleotide, ATP. ATP is also a nucleotide. It's a derivative of the nucleotide. It's a nucleotide, right? The ATP, we all know that ATP is the energy currency of the cell. That is my next topic, right? So here, this is ATP, right? If you see here, ATP is also a nucleotide. You know that it's an energy currency of the cell, right? Without ATP, we cannot do anything without ATP, right? The energy is stored in the ATP, right? In the bonds of the ATP, right? So whatever processes are happening in our body, right? ATP is the one which provides energy to the various processes in our body, right? For example, last semester we spoke about muscle contraction or nerve impulses, right? So ATP is the one which will supply energy, okay, in our body. And ATP is also a nucleotide, okay. So in this nucleotide, we have an adenosine as a base, adenine as a base. Okay, as you can see here, there's adenine as a base. Okay, there's also sugar present in the ATP. Okay, the sugar is the ribose in the ATP. So structure of ATP is also very important. Okay, so ribose is the sugar in the ATP, or ATP is having 1, 2, 3 phosphate. So that is why the word adenosine triphosphate. But does that mean nucleotides can be only monophosphates or triphosphates? No. A nucleotide can also have only two phosphates, as you can see, ADP. Okay, ADP is a nucleotide which has only two phosphates. Are we okay till now? The bond energy and everything about the ATP that we will discuss in the later chapters when we'll discuss the biosynthesis. But here you just need to know the structure of the ATP. That is it. Okay. Now, if you see here in the ATP, okay, the bonds in the ATP, okay, if you see here, just remember the names. That's very important, right? So in the ATP, if you see here, it has one phosphate ester bond over here. So this is a phosphate ester bond in the ATP. This is a phosphate ester bond in the ATP, right? Which is present between the phosphate as well as between the 5' carbon of the sugar. And other than that, if you see here, let me erase all this. Other than that, if you see here, it also has what? How many anhydrides? It also has two phosphoanhydride bonds, this one and this one. So this is like, I'm going to make a star over here to show you that, let me make it with the yellow color, to show you that the energy is stored in these two phosphoanhydride bonds, which we will learn in the upcoming chapters about the energy liberated. Okay, right now just remember the structure over here. So the question says that how many anhydride bonds, phosphate anhydride bonds are present in the ATP? What will you say? You will say that ATP has two phosphate anhydride bonds, and at the same time it has one, it's connected to the sugar. Like these kind of questions you can expect. Okay, at what position is, or at what position is, okay, if I give you the structure of the ATP, let's say I give you the structure of the ATP, okay, and just ask you to name it, what is it? So the moment you see adenine over here, okay, the moment you see a ribose sugar over here, maybe I can give you a deoxyribose. So you should know that no, ATP has a ribose sugar over here. It doesn't have the deoxyribose. So just to confuse you, I might give you deoxyribose, for example, right? So I might not give you adenine over here, right? I might give you a single ring over here, okay? I might give you, a pyrimidine over here, like thymine or uracil, right? So you should know that it has a purine. It doesn't have a pyrimidine. As I said that yes. See, I cannot say that don't remember the structures, because when I, for example, me or Mr. Hindavi, if they give structures, you are in a big trouble then, right? So you don't have to know the structures one by one. Where is N? How many double bonds? But if I give this structure, I can definitely give this structure, saying asking you what kind of, which of the following shows us the ATP in, in the following options. So don't know it bond by bond, but yes, being a biochemist, structures are like part and parcel of the biochemistry, right? So yes, you should be able, the way we had in sugars, for example, you should be in a position to know that, oh, this is a glucose, this is a fructose, this is a pentose sugar, this is a triose sugar, right? So these guys, similarly in this chapter also, you should be, I cannot say that don't know the structures. No, it's not completely no. You now know that what was the paper, what was there in the midterm one, right? So it was not totally structure based, but at the same time, yes, we had, for example, if I tell you today that don't memorize any structure, just in case. Tomorrow, if I give any question about just identification of the structures, at that time the student will tell me that no, you said don't know don't memorize the structures. So we never tell any student not to memorize the structures, right? But as I'm lecturing, I just try to brief you, how you should, know a particular question, okay, what is expected, to so that get an idea, okay. So yes, structures will be there, but you don't need to know the structure. Okay, here we have an NH2, then we have an N, then we have a the alternate double bonds are over there. No, that thing you can be a little relaxed about it, but structures are there. Okay, Nana? Is it clear? Are you guys tired? We didn't even finish the 1/4 of the chapter. I'm actually going very slow, but that's okay. As long as you are comprehending the information, that's okay with me. So, are we okay till now? I mean, I'm not going fast. Everything is comprehensible right now, right? You all can understand it. So just let me know if there's something which you don't understand, or you can just definitely stop me in between. Okay, if you don't understand anything. Okay, Alhamdulillah. Thank you, dear. Thank you, dear. Okay. So, if, for example, if I give you the question like identify the ATP, you as a biochemist should know that oh, ATP has. She said that ATP doesn't have the pyrimidines. It has purines. So that means if you see here a single ring, oh, that means this is not an ATP, right? Or you should know that oh, she says that ATP has a sugar, but the sugar is a ribose. It's not a deoxyribose. So if you see here like as an H over here, so it should come to your mind that oh no, it can't be ATP. It is a ribose sugar. It's a deoxyribose, but in ATP we have a ribose sugar. Okay. Or if you see here just only two phosphates over here, it should come to your mind that the ATP is a triphosphate. So adenosine triphosphate, right? So that is how you need to memorize about it. Okay. And if I ask you about what are the energy bonds present in the ATP are your phosphate anhydrides bonds. Every time when we say ATP gives energy, okay, so this structure will form a base for all of you, when because we're going to start about metabolism, the biosynthesis in the upcoming chapters, right? So when we talk about okay, ATP, the phosphate bonds are broken. So at that time, imagine that these two bonds are broken. That's why I made them yellow to show that they are they liberate the energy. Are you okay? Okay, now if I ask you a question, let me see what have you understood the concept. If I ask you a question, okay, what is the difference between the nucleotides in the DNA and the nucleotides in the ATP? How will you answer? I repeat, what is the difference between the nucleotide in the DNA and nucleotide in the ATP as a nucleotide, for
The
example?
phosphate groups,
The phosphate groups, very good. In DNA or the RNA, how many phosphates we have?
only one, and
One.
in ATP
Wonderful.
we have three phosphates.
Very good. The basic difference is in the DNA, you have only one phosphate, monophosphate, while as in the ATP, you have the triphosphate. Very good. And then the other differences, like in the DNA, we have the deoxyribose sugar, and here we have the ribose sugar. Okay, that is about it. So we did all this. So the nucleic acids, DNA and RNA are the polymers of the nucleoside monophosphate. So I just put in here so that you don't forget this. They are the monophosphates, whether it's in the DNA or in the RNA. They are not triphosphates. Okay. And remember, nucleotides can be diphosphates as well as biphosphates. ADP is an example of a biphosphate, and AMP is an example of a monophosphate at the same time. And then what are the other nucleotides? That those are, you don't have to know them here in this chapter, but yes, when we will discuss biosynthesis and other thing, you'll see that we'll be frequently using the NADH, and the various other coenzymes. Okay, so those are also nucleotides, but you don't have to know all that in this chapter. That as we'll discuss the biosynthesis, so I'll explain it in there. Okay. So then I brought a picture here so that now all the bonds. Okay, this is a nucleotide. Okay, this is a nucleotide. Is it a deoxy ribonucleotide or just the ribonucleotide, guys? We're looking at the structure. What is it? Is this nucleotide present in DNA or in the RNA? How can you
DNA.
it's present DNA? Because at the position number 2, I
Um,
don't have
we have
OH, right?
oxygen.
That should give you an idea, that this is present in the RNA, not in the DNA. I mean, this is in the DNA, not in the RNA. I mean, yeah, right? And in this picture, I'm just trying to show you that this is a base. A nucleotide has a base. A nucleotide has a ribose sugar, and a nucleotide has a phosphate. And I'm trying to explain you that the bonds, just going to repeat it once again. In the nucleotides, the sugars and the bases are connected by via the glycosidic bonds, which can be N9 of the purine and anomeric carbon 1 of the sugar. And if it's the pyrimidine, it will be here, what, N1 of the pyrimidine and the 1 of your sugar. So this is a glycosidic bond over here. Okay, and then how is phosphate connected to the sugar? Phosphate is always connected by the phosphoester bond, right? Which can be connected at the position number 5, or it can be also connected at the position number 3. This is something which is very important, which you all must know about it. So there is one more picture here to differentiate between a nucleotide and a nucleoside. There's no phosphate over here, and there's a phosphate over here. Right? There's one more summary over here: nucleoside base plus sugar, nucleotide base, sugar, phosphate, right? And a nucleic acid, it contains a chain of nucleotides. Many nucleotides together are present in the nucleic acid. Okay. What is the monomer of the nucleic acid, guys? The monomer of the nucleic acid is it is a nucleotide, isn't it? This is a nucleotide. The same thing what we discussed. What is the polymer of a nucleotide? Nucleic acids, many nucleotides together, right? If you see here, this is a polynucleotide. Okay. So this is a polynucleotide, right? Wherein you have, look at this, you have a phosphate, sugar, and base. This is your one nucleotide. Again, phosphate, sugar, base, another nucleotide. Phosphate, sugar, base, another nucleotide, right? Okay. So these are many nucleotides together over here. I will repeat. I said in the, you mean to say in terms of the sugar? Eileen, are you talking about in terms of sugar, Eileen? Right? If you see here, this sugar doesn't have OH over here. There's no OH over here. Right? If it doesn't have OH, if there was an OH over here, then I would know that this sugar is a ribose sugar, isn't it? So that means this sugar is present in the RNA. Okay, but since it has no oxygen, so what do I know? If there's no oxygen over here, that means this sugar over here is my deoxyribose. Okay, what does that mean? That means this is a nucleotide which is present in the DNA, not in the RNA. That was my question basically, and that's what, you know. Is it clear, Aileen? Sugar is going to tell you that whether it's a nucleotide which is present in the DNA, or is it present in the RNA, isn't it? That's going to help you to identify is it a nucleotide present in the RNA, is it a ribonucleotide, or is it a deoxyribonucleotide? Okay. Shall I move to the next one? Structure of DNA. Okay. So was it all clear till now? I mean, is it okay till now? Is it like, how did you find this chapter till now? Yeah, Mohammed, that we're going to talk about once I discuss the structure of the DNA. I'll discuss the phosphodiester bonds, you know, here. No. Can we say nucleotides contain phosphodiester? Mohammed, I just would suggest you to wait here. Okay, wait for some time. I'm going to explain about the phosphodiester bonds as I explain the structure of the DNA. Okay, so glycosidic bond, Mohammed, you have to remember glycosidic bond. Okay, okay, thank you, Mohammed. Thank you for the compliment. So you see, for the glycosidic bond, you have to remember that the glycosidic thing. Only one thing you have to remember that the see phosphodiester bond has nothing to do with the glycosidic bond. I don't want you to get confused about it. So that is why I brought this picture here. Okay, because I just want you to be very clear. Simply remember the base and the sugar in the nucleoside or in the nucleotide. They're connected by the glycosidic bond. So remember that. If the question says that, how is your base and sugar connected, right? Remember that it is always a glycosidic bond, Muhammad. So glycosidic bond has nothing to do with the phosphate. Okay. Is it okay for everyone? So it's the beta N glycosidic bond which connects your base and the sugar. And then I of course told you that if it's a pyrimidine, it's N1 and purine, it's N9, right? Is it okay for everyone till now? Is it clear, Mohammed, till now? Glycosidic bond has nothing to do with the phosphate. Are we okay till now? Okay, great. Now, when we talk about the ester bond, I didn't say phosphodiester bond. I just said the phosphoester bond, or simply use the word ester bond. Okay. Now the question says that how is phosphate connected to the sugar? See that sugar is kind of a poor person who is stuck up between two people. Think it this way. You can memorize it this way. Sugar is a poor person, who is squeezed between two people. From one side, base is squeezing it, okay, using the glycosidic bond, and from the other side, phosphate is squeezing it, okay, using the ester bond. So phosphate is connected to the sugar by the ester bond, and that ester bond can form at the carbon number 5, or that ester bond can form at the carbon number 3. Just remember that. Don't phosphodiester bond, I will explain you when I will discuss about the polynucleotide chain. When we have many nucleotides together. Clear for everyone? Okay. Okay. Now let's move to the sugar by the anomeric carbon. Absolutely. The C1. Yeah, it has to be always C1. But for the base, it has to be N1 or the N9 for purines and the pyrimidines. Okay. And for the sugars, if you remember, I told you that the sugars, they have to be the furanose sugars, the 5-membered sugars. They have to be the diisomers, right? And the configuration has to be the anomeric carbon, which is a beta form of the sugar. You know the alpha and the beta form now of the sugar. There was a question also about the beta and the alpha form of the sugar, right? So it is a beta form of a sugar, okay, in the nucleotides. Now, the structure of the DNA, okay? So the structure of the DNA, it has a primary structure, it has a secondary structure, and then it has a higher order structure. The way you might have, studied in proteins, right? The primary, the secondary, the tertiary, the quaternary structures. So DNA also has a primary structure, secondary structure, and then the higher order structure. Okay. So when I say primary structure, if someone will ask you about the primary structure, what do you know from the primary structure? Primary structure of DNA simply tells us the sequence of the nucleotides. That is it. Okay. So primary structure of the DNA simply tells me how are your nucleotides arranged in the DNA. We always begin with remember this is again an important point which we will use in the replication and the transcription. We always begin with the 5' terminal, okay, in the DNA. And it's like, if this is my DNA, so one strand is 5', okay, and another strand is 3', right? So and the other strand of the DNA will be always anti-parallel, right? So this strand will be my 5' end, and this will be my 3' end. Are we okay with for everyone? Is it okay with everyone? Anti-parallel. The two strands of the DNA are anti-parallel. It's like, you're driving a car, okay? There's a divider in between, okay? Some of you are going towards the Alpha SL University, and others they're finished with the classes, they're coming out of the Alpha SL University. That is the anti-parallel. That's how you can remember it. Okay, so 5' 3' and 3' 5'. It has two ends, the way we spoke about two ends, in the proteins, right? The C' terminal end and the N' terminal end. Similarly, here we talk about the 5' end and the 3' end. Is it clear to everyone? Okay. Guys, are we connected? Nobody's answering? Okay, yes. Okay, thank you. It takes time to write. Yeah, definitely. Yes, okay. Now I want you to pay attention. If I give you a sequence like this, the way I have a sequence over it, adenine, guanine, thymine, and I ask you, identify the 5' and the 3' end of it. Doctor, from where 3' 5'? One, okay, it's about one strand is 5' as you can see over here. One strand is 5' and the other from 5' to 3', and the other is the opposite. This is like from 5' to 3' like this, and the other goes from the opposite 5' to 3' like this. We never say 3' to 5'. We always say 5' to 3'. Okay, we go from 5 to 3. Okay. Now, if I give you a sequence like this and ask you, okay, tell me, I give you a sequence like adenine, guanine, thymine, adenine, cytosine, a long sequence, and I tell you, tell me, okay, which is the 5' and which is the 3' end, right? So you will never begin with the 3' and 5'. You'll always begin with, you say that this is the 5' end, okay, and this is the 3' end. You will never say the other way around. You will never say that this is my, you will never say that this is my 3' end and this is my 5' end. You have to always read it from the 5' to, so this is the wrong way, then your question will be totally wrong. You will always start with the 5', and this you have to remember forever, because your replication, your transcription is based on, from 5' to 3', and whenever you have to read the DNA strand, you will always read it from the 5' to 3' end. Okay. So I think we'll stop over here, because even I have a next class, and I'm sure that you all must be tired now. Is it? So this was just the beginning. As I said, that we didn't even finish 1/4 of the chapter, but that's okay. Slow and steady wins the race. So do you have any questions for me? If you have questions,
Doctor,
still have 6.
could you
Yes,
repeat the last part, please?
Yes, dear. I will repeat one. I didn't even finish it because I wanted to show you, a diagram as well. If you see here, this is what I mean. Okay, this is my 5' end. Okay, and this is my 3' end. Okay, so if you're given a sequence like thymine, if you see it is a thymine here, cytosine here, right, and guanine here. So thymine, you will read it as 5' end. You will always start with the 5' end with the 3'. We never say 3' to 5'. For example, whenever you have to read a particular DNA sequence, you will, the first base is always a 5' end. Remember this way. Goodbye, everyone. Is it clear, Ran, what I'm trying to say? So this is like, I didn't even finish this topic yet, okay? And this will be continued further in our Wednesday's class.
Yes, it's clear. Thank you, doctor. Have a nice day.
You will read it always, for example, when you read ABCD, we never say Z, Y, X, right? We always read ABCD as an A, B, C, D, E, F, G, H, right? Similarly, when you have to read the DNA strand or any bases in the DNA strand, you will read it from 5' to 3'. That's what I mean basically. Okay.
Thank you, doctor. Have a nice day.
Thank you, everyone. All the very best. Have a wonderful afternoon. Okay, and let's meet on Wednesday, inshallah. Not meet exactly. Let's have an online class on Wednesday, inshallah. Bye, everyone. No. You don't need to write your names, Yusuf. Your attendance is automatically recorded. As you log in to my account, I get an Excel sheet wherein I have all your names there. Okay. Thank you, everyone. Yes. Bye, bye, everyone.